So I would like to introduce Ming-Chia Lee. The title of her talk is Progenitor Expansion and Competence are controlled by Lsd1, PRC2, and non-coding RNAs.
So first, I want to thank the organizers for giving me this opportunity to present my work. So a precise control over the differentiation onset is really key to tissues and organs. For tissues or organs to reach and maintain their target size in a healthy condition. And misregulated onset of differentiation will lead to detrimental cellular consequences. For example, a premature onset of differentiation would generate too few of cells and compromise tissue integrity. On the other hand, a delayed onset of differentiation will lead to tissue outgrowth, which could be the first step toward tumorigenesis. It has been generally assumed that the timing of differentiation is controlled by the appearance of external cues. However, it is less clear whether intrinsic program can be actively controlling individuals' progenitors responding to external cue. So that regulations on the cell contents could be the key to control and determine the onset of differentiation. And basically, my project is set to understand the molecular basis of progenitor competence acquisition by using the ovarian follicle cell lineage, where the follicle progenitor cells will undergo 9 mitosis before they switch to endocycle. And this N to E transition is known to require Notch activation. Before our research, it has been generally assumed that the appearance of external cue, the delta, the Notch ligand, is really what triggered this transition during this, after this rapid mitotic phase. However, not only that the expression window of delta is earlier and wider than the ME transition, but also I discovered that these early progenitors, they are not competent to respond to external cue, the Notch activation. So when challenged with the expression of Notch intracellular domain expression, shown in the red here, the early progenitors, they cannot undergo premature ME transition. Rather, they keep dividing and in the end generating the comparable number of follicle cells compared to control, suggesting that the contents has to be acquired from early to late progenitors, and presumably from some type of intrinsic program. Our system is particularly powerful of understanding the competence acquisition, partially because the follicle progenitor cells will undergo 9 mitosis stereotypically. So that simply by counting the number of cells, we can determine the timing of differentiation. And we can further infer the timing when progenitors acquire their competence. For example, the reduced number of cell will indicate a precocious competence acquisition, while an increased number of cells would suggest a delayed competence acquisition. Moreover, by using this USGP variegation system reported a couple years ago, we were able to trace and tracking the epigenetic changes occurring during this rapid progenitor proliferation phase. So basically, we know that the reduced epigenetic plasticity will be manifested by the suppressed variegation pattern, while the elevated epigenetic plasticity will lead to enhanced variegation compared to control. And we discovered this tight association between the timing of differentiation and the variegation pattern, suggesting there could be an epigenetic control over the competence acquisition process. As you can see, the precocious ME transition is associated with a much less variegated pattern, while the delayed transition is correlated with enhanced variegation pattern. Therefore, to look for this epigenetic regulator, which can be presumably modulating the progenitor competence excision, we look for the modifiers of this variegation pattern specifically. And that's how I identify Lsd1, the lysine-specific demethylase 1, as a suppressor of variegation. And indeed, it regulates progenitor differentiation as a F3K4 specific demethylase. So as you can see, for progenitors has mutant a copy of Lsd1, they show reduced epigenetic plasticity, and you can see they are less variegated compared to control. And I also discovered this nearly linear correlation between the number of follicle cells to the Lsd1 dosage, suggesting the timing of differentiation here is really sensitive to Lsd1 function. So basically, for progenitors has reduced amount of Lsd1, they undergo premature MU transition and generate fewer cells. On the other hand, when they have excess Lsd1, they undergo delayed MU transition and generate more cells. And this result basically indicate the potential Lsd1 function in suppressing competence excision. And the idea can be further supported by the Lsd1 expression pattern. As you can see, Lsd1 expression is almost absent after MU transition. And you can see there is a gradual reduction of Lsd1 expression from early to late progenitor cells, suggesting it could be underlying the derepression of competence excision, which is required for the MU transition. We know Lsd1 does not function alone in this context. Rather, it binding to its canonical binding partner co-rest to be a F3K4 specific demethylase. We know this strong interaction between Lsd1 and co-rest can be reliably detected by a co-immunoprecipitation from ovarian lysate. And interestingly, this strong interaction is RNA sensitive, suggesting some type of RNA can be associated with Lsd1 complex and modulates function. Therefore, I perform Lsd1 specific RIP-seq to identify potential non-coding RNA components that associate with Lsd1 complex. And here's a list of 4 candidate genes I identify. So they show, compared to IgG pull-down control, they show 15 to 50-fold enrichment. And for today's talk, I'll mainly focus on the first 2 candidates, which are longer in their length, including CR32205 and PNCR009. So indeed, by genetic assay, we found both non-coding RNAs regulate progenitor differentiation. So as you can see, very similar to Lsd1 heterozygous, the CR32205 allele leads to suppressed variegation. And interestingly, the PNCR009 RNA leads to enhanced variegation. And they also lead to corresponding changes in the timing of differentiation. So while the CR32205 heterozygous showing this premature MU transition phenotype, the PNCR009 leads to the delayed MU transition. And this opposite phenotype of these 2 non-coding RNAs can be confirmed by looking at the heterozygous mutant generated by FRT recombination. And we know these 2 non-coding RNAs modify progenitor expansion in an Lsd1 dependent manner. So while the PNCR009 can rescue the premature MU transition in the 1X Lsd1 background, the CR32205 heterozygous mutant can speed up the originally delayed MU transition in 3X Lsd1 background. And the interaction between CR32205 and Lsd1 can be further confirmed by the in vitro assay, where the in vitro synthesized and biotinated CR32205 can pull down both the Lsd1 and co-rest from ovarian lysate. But to our surprise, it also pulled down another really famous factor, the EZ, which is F3K27 methyltransferase. It's a key component of polycomb repressive complex 2, the PRC2. And this binding result would suggest a potential involvement of PRC2 in regulating the progenitor expansion too. And indeed, when we look at all three co-components of PRC2, the SUS12, ESC, and EZ, they all show phenotype as a suppressor of variegation, as you can see right here, compared to control. And they also affect the timing of differentiation. While the SUS12 and ESC show a mutant leads to premature MU transition, the EZ leads to delayed MU transition. And this specific function of EZ actually is depending on Lsd1 again, because when we look at the double heterozygous, it actually rescues this delayed MU transition phenotype back to wild type. So based on the data we have so far, we believe in early progenitor cells, Lsd1 function as an epigenetic eraser, and it constantly removes the methyl group from the F3K4 residue to keep it from being saturated, so that to keep the key loci, such as notch target genes, at low competence state. And the involvement of PRC2 likely make the regulation even more complex. So among target or key loci, such as notch target genes, they may be either bound to Lsd1 or PRC2 or both. For example, like the key notch activator hindsight could be repressed by either or both of these two complexes. And this modulation of Lsd1 and PRC2 is likely being modulated by non-coding RNAs. And we are currently trying to understand how the non-coding RNA is modulating this complex, either by regulating Lsd1 stability or function as either regulatory or scaffolding role in modulating to coordinate the function of PRC2 and Lsd1. And we believe in the late progenitors, the reduction of Lsd1 expression is key to lead to the repression of key loci, so that they can now respond to notch activation and initiate differentiation. So now I would like to thank Alan Spradling to be such wonderful mentor. And I also want to thank really great supporting staff in Carnegie, and also the Fly community and the funding source. Thank you very much.
This time we do have time for questions.
Is there any similarity between the two non-coding RNAs? Do you think that they pair in any way?
That's a really good question. So I didn't have time to go into. They are very, very similar in their sequence. And so they are like 80% similar in their sequence. So it's possible, because they show this opposite phenotype, there definitely could be some interaction between these two non-coding RNAs. But we don't have like clear answer yet.
So when I think about polycomb-mediated repression, I think about it as sort of a permanent thing, or for the rest of the lifespan. But your model suggests that it's being reversed, and so is that a common thing? Are there many models for that? And how do you? What's the mechanism for reversing polycomb silencing?
Yeah. So the interesting thing, if we look at just the F3K27, F3K27 trimethylated staining, you can see it's very strong in like early. Just before the MU transition, it's very strong, but it's almost absent afterwards. But for that, because we don't really have a resolution, but it can suggest there's the function of PRC2 there. We don't really know what's the dynamics of it at this point, but I think we can see the consequence of it. So they definitely function at like before MU transition. But that'll be interesting to know how like dynamically being regulated. Yeah. Okay, thank you.